So I'd like to introduce our first speaker for the session, Dr. Susan Gibson-Koo, who will be presenting on the pathology and blood biochemistry of juvenile lates calcifer in diets containing mycotoxins, histamines and rancid fats. Welcome to the stage, Susan. And I'd just like to remind our speakers for this session that we'll give you a bell at 5 minutes. And then it's up to you, I guess. Thank you, Susan.
Hi, I'm Dr. Susan Gibson-Koo. So if you're all seated, I'll start. I'm from the Tropical Futures Institute of James Cook University based in Singapore. And today I'll talk to you about what happens if you investigated fish that were put on diet. It was not by, it was not intentional. And so what we did was, this was based on fish kept in RAS, recirculating system or experimental recirculating system, and we were running a trial on finding alternative proteins fed on recycled kitchen waste, when we found that our control fish were doing very poorly. So poorly that we decided to terminate the experiment early and intending to start again. And we investigated why the fish were doing poorly. So you can see that the growth performance was bad. The food conversion ratio was very much greater. Just to give you a little bit of background, ABS3 is a commercial diet. These are control diets with an alternative protein diet, but we only investigated the control diet to see why the fish were doing poorly. And the one fed the fish meal protein 40 was a diet we formulated as a control diet with 40% fish meal. And it was meant to be doing better than the alternative protein diet, but they were not. Their feed conversion ratio, after accounting for mortality, you can see the survival rate is very small, at 70% or less. Food conversion ratio was greater than 2. The growth rate was very small, and what was surprising was that the fish were eating much, much less than the commercial diet, for a control diet that we expected to have normal consumption. And so we sent our feed at this point in time, we did not know that there was anything, or we weren't sure what was the cause of the poor growth performance. When we sent the feed for analysis, we found that they were contaminated with a lot of aflatoxin, and different kinds of other mycotoxin like zearalenone and fumonisin, and also that they have high peroxide values, meaning that they contain, fish usually have a lot of high protein diet, high protein unsaturated diet that is very easily oxidized in warm temperatures, and so they were getting rancid, and they were also high in histamines. And histamine tend to be from, if the feed was formulated from fish meal that were already spoiled. In other words, that some of the amino proteins had that got degraded down to histamines. And histamine is like, you know, the chemical that controls inflammation and allergies. And so we look at histology, and we also did some blood work. Even though blood biochemistry is not commonly done in fish, we did not have normal reference value, we decided to do and compare and see what we find. And you can see that the liver on the left is very dark colored, and that's because they have lost their fat and glycogen reserves. Fish, they are eating really well. We have a lot of fat and glycogen reserve, so they look like the one on the, I have to see whether they are the same as the one I'm looking on the left, right? The lighter staining one. So that one is a normal fish, and that was the one that was fed on the diet, the commercial diet ABS3, and the other one was with the dark looking liver was the one fed the fish meal 40 diet that we formulated, right? With the mycotoxins and other types of aflatoxin, fumonisin, and all the mycotoxin in low levels, I might add, not really high levels, right? And you can also see the star. Normally when you see a section of the liver, you might see in one particular spot, you will see one section through the bile duct. You can see multiple bile ducts, right? And so this suggests that the bile ducts have proliferated. They become tortuous. You know, like if a duct is like a pipe, it's become very tortuous and convoluted, so that when you cut a section through it, you see many, many sections. Right? This suggests that they are reacting to something. You know, the liver detoxify things, right? And they make them into products that can be excreted either through the kidney afterwards, or maybe even through the bowel. They cause them to become increase in length. So that's why you can see the difference. It's not bad though. You've seen chickens, they are fed food with a lot of mycotoxin. They are much, much worse than this. And so maybe this is because these fish were only on this diet for 5 weeks, right, when we terminated the experiment. So it's not that bad yet. That's what we found. And we did more spatial stain. The top two images are a section through the spleen. And in this particular spatial stain, red blood cells were stained yellow. And you can see that the fish that were fed the ABS3 commercial diet, which has less mycotoxin, had more red blood cells, right? Of course, it's red blood cells, but in this special stain, they're staining yellow. You can see there are more blood cells than the other one. They are fed FM40. And when we did Persian blue, which is a stain for iron, you can see that the fish that were being fed the FM40 were accumulating a lot of iron, which stains blue. Normally, even in normal healthy fish, our red blood cells recycle, right? And you will get some blue color in the spleen, but you can see it's much, much more in the FM40, suggesting that the blood cells were turning over faster, right? Usually due to a chronic disease, can happen in viral disease, or even in other diseases maybe, where any kind of toxin causes the red blood cells to die faster than usual. And we did blood biochemistry, and we chose the vet scan because, you know, fish are kind of small, and it's very hard to take blood like from a pig, a chicken, or a cow. This one only needs 100 microliter or 0.1 ml of blood kept in anticoagulant like heparin, and we put them into this. You can see the rotor with the red beads in it. So you feed the 100 microliter into this rotor, you put it into like a cassette, you know, like a CD player. You put it in, and in 5 to 10 minutes, you get a range of all these blood biochemistry, which is like the panel that you get when you go to the doctor, you know. They'll tell you you have liver disease, blah, blah, and so forth. But what we found that in the fish fed the FM40, you can see that potassium is very, very high at 8 millimoles per liter. Potassium is usually very regulated in the fish, so it shouldn't be high. So we wanted to investigate why. So we compared with the feed analysis, right? And so figures that a lot of the mycotoxins are cytotoxic. They will cause any cell to die. They don't specifically choose the red blood cells. It just so happened that the red blood cells is one of the fish, one of the cells that also has a high turnover, right, or dies. So that's why they release the potassium into the blood. So you can see the high blood potassium, and you can see the high iron. Remember this picture where you have the high iron that's accumulated. These are aggregates or groups of macrophages. They scavenge the iron because in nature, iron is a very scarce compound. So when you lose blood, the body tends to reserve the iron for you to use it to make more red blood cells in the future. And then we look at the kidney also. You can see, maybe most of you are not used to looking at sections of the kidney. You can see where the star is. They are anything that stains pink in this H&E or hematoxylin and eosin stain is protein, right? And this suggests that the kidney is being damaged and you are losing protein in the kidney, right? And when we look at the feed in this one, this is the feed fed the commercial diet, the high histamine. Literature has it that, these are from literature that people do mainly from medicine, you know, like it causes, it is degraded in the kidney and it can cause damage in the kidney such that you lose protein, right? You shouldn't have protein in your kidney unless you were a catfish. Sometimes they do that. during seasons when they are building nests. They have protein in the kidney to build the nest. But it shouldn't happen in other fishes, right? So that suggests that the kidney was being damaged by the histamine. And then you can tie all this back to the poor growth performance, the poor food conversion ratio, and so forth. And you can see here we did another special stain called Marshall Scarlet Blue. It is to look for blood clots, right? When your tissue are damaged, you release a lot of broken cell beads into the circulation, right? And that triggers the coagulation cascade and causes blood clots. It then tells you that even when, sometimes, right, when you look at pathology, it has to be really bad before you can see lesions. But if you do this special stain, even when they are minor lesions and they are causing blood clots, you can see them. They are the ones that stain red around the blood vessels, in the liver, and then also the kidney. So it gives you confidence that there's some kind of tissue damage going on, and helps you to explain that, I found mycotoxin, I found histamine in the feed, and this is causing a pathology effect, and it explains the poor growth performance. And also it affects the immunity. We found that usually in baby fish or juvenile fish, you should see 60 to 70% of the profile of the blood cells as lymphocytes. In these guys, there were only 20%. So it shows that it's having effect on growth performance because of all these issues, degrading the red blood cells more than usual, and then also depressing the amount of lymphocytes that is part of the immune system of the fish, right? And so this fish might be more prone to anything, right, including diseases. So again, you see here, I forgot to tell you that we also found a high calcium, suggesting that the high calcium is because of the kidney damage, right? And also one of the, I didn't look at it before. You can see the oxyzelenol. That mycotoxin is also called vomitoxin. It makes you vomit, right? So you can imagine that if it's high amounts, maybe if the fish vomits, that might explain why there's low feed intake. Because usually we feed and then we measure the leftover feed and then we see how much the fish is eating. Okay. So the key findings in this one is that it's very common, right? You don't think about it because you don't investigate and you only see what you find. That we find that a lot of the poor growth performance, right? Maybe even other diseases that are present all the time, but why are they showing up in the fish? Could be because of underlying mycotoxin in the feed, which is very common, which causes kidney or liver disease, and then also increased turnover of red blood cells or hemolysis and iron storage. How are the management? The good news is that if you find it out early and you remove the feed and replace it with better one, within several days, the mycotoxin level in the flesh and in the tissues reduce remarkably. So you can find early, you can stop the poor growth performance and the poor immunity if you correct the feed. And I also wanted to show you these micromarkers. I've been trying it out. You only take a drop of blood, you put it on a spot, with the circle in the filter paper, and you send it to Innovet, and they can actually check the blood in there for mycotoxins. And they usually check down for growth products, and we have sent them several times. And very often we find products of mycotoxin in fish blood, even when they are apparently doing well and clinically healthy. So this concludes my presentation. And I hope that more people will do things that people say that there's no reference value, so there's nothing to compare to. You can see if you compare between things, and if you use it with an established tool like pathology, where there's a lot of information on what you see under different disease conditions, you can put the two together, and you can see the importance also of checking the feed for mycotoxins. And then it can explain why you're getting poor growth performance in fish. Thank you.
Thank you, Susan. We've got some time maybe for a quick question. I like that emphasis on looking at your fish and the feed. So I'm doing some some diagnostic work to see why the animals are actually not performing, and I love pathology in there. That's great. Thank you very much, Susan.
Thank you.
We call to the stage our next speaker, Raja Suwanathan, evaluating the suitability of fish cell lines in toxicity assessment of metal oxide nanoparticles. Thank you.
Very good morning. Today, this may be the critical part of our ongoing study on the metal oxide nanoparticles for the various applications in our study. So, if you are seeing the OECD guidelines of 2003, so the guideline says that live fishes can be used for assessing the toxicity in the aquatic environments. In addition to the fish embryos can be used, as well as fish fingerlings can be used. The same guideline says that we have to explore the alternate to use the live fishes for the toxicity studies. So the same OECD is recommending that RTG gill cell line can be used for assessing the cytotoxicity of the chemicals in the worldwide. So if I seeing the EC50 value derived from the cell line, it is to be an excellent agreement with the LC50 value derived in the fishes for most of the chemicals. So this that's why OECD is recommending the use of fish cell lines for assessing the toxicity in this one. So this is the global status of fish cell lines available. Maybe as on today, if you are going to the Cellosaurus, the encyclopedia of cell line, more than 1,500 fish cell lines has been documented worldwide globally. So when there is 1,500 cell lines available, why OECD is recommending only one cell line for using one cell line to be used for the cytotoxic assays? The main reason what we are thinking that, so this RTG gill cell line is developed from the gill tissue of rainbow trout. So the gill tissue which is having the direct contact with the aquatic environment, so the cell line developed from this gill tissue may be a suitable cell line for assessing the toxicity. In addition to that one, the most important thing what we thought, so more experiments were being conducted by using these cell lines worldwide. That's why this RTG gill cell line was recommended by OECD. So if you are seeing the Indian scenario, so there is National Fish Cell Line Repository. The biggest fish cell line repository globally is established at NBFGR Lucknow in India. So this is maintaining more than 84 cell lines. This is the set of the cell lines. So we thought that why these cell lines cannot be used for assessing the toxicity assays. Suppose if you are seeing the application part of these cell lines being used in India, it is mainly used to isolate the finfish viruses in India. In the morning, Dr. Sagu sir, he was telling that more than 12 finfish viruses has been documented after the NASPAT program. So this is the main reason is, in addition to NASPAT program, the availability of cell line in India is the main cause to isolate the viruses in India. So we thought, so this is the time to explore the finfish cell lines, which is of Indian origin, which can be explored for assessing the cytotoxicities. It is having more benefits than using a single cell line. So because the more benefit what we have thought that, so the study on species-specific response on the pollutants can be explored when we are using more cell lines instead of a single cell line. So why we took the metal oxide nanoparticle for this study means this is an ongoing study in my lab. So if I see the global market, it may be a $53 million, starting from cutting across all the industries like electronics to cosmetics. So what the disadvantage here means, when the nanoparticles is maybe excreted into the aquatic system, it can hinder the maybe food web in the aquatic system. It can affect the algae, it can affect the invertebrates, then it can also cause disease in the fishes. That's why this study has been taken. So this is our ongoing study in my lab. So we are preparing the metal oxide nanoparticles. Here I am discussing about the zinc oxide nanoparticle prepared and its uses. So after the preparation, we used to characterize the nanoparticles. Then this nanoparticle will be assessed for its antimicrobial activity, wound healing ability, as well as the anti-cancerous activity. So in addition to that one, we are doing the cytotoxic assays by using different cell lines. In this study, the fish cell lines, use of fish cell lines in assessing toxicity, which will be compared with the human as well as the rodent cell line. So here we are using MTT assay as well as Risasurin assay for our comparison. So here the fish cell line, we are mainly we taken only 3 cell lines of 3 different tissue origin. One is Horobrachus brachysoma fin cell line, another is Oscar spleen cell line, another is goldfish gill cell line. These 3 cell lines we taken for analyzing the cytotoxic activity by using MTT assay as a Risasurin assay. This research was compared with the 2 cell line, one cancerous cell line, MCF cell line of human origin, another somatic cell line of L929 of rodent origin. So the results what I am going to present, it is only results of HBF cell lines. Due to the time limit, I am representing only the results of HBF cell lines. So, as usual, we prepare the nanoparticle. The nanoparticle has been characterized by different means by using the UV spectra as well as DLS and SEM. So, the nanoparticle what we prepared, it is confirmed that it is having all the characteristics of nanoparticle. So, these nanoparticles we used to test for the cytotoxic effectivity. So, the top panel you can see the yellow panels. It is the 96-well plate of HBF cell line, toxicity effect on HBF cell lines by using the zinc oxide bulk particle as a nanoparticle. The bottom line you can see, which is in the green panel, it is the composite of nanoparticle coupled to the chitosan. When we are using chitosan, the toxicity was reduced like anything. So when we are not using the chitosan with the nanoparticle, the toxicity is more. So here we can see, this is the main finding I would like to discuss with you. When we are using the fish cell line to assess the MTT assay, the color development which is supposed to be get, because when we are doing the MTT assay, the yellow MTT will be oxidized by the mitochondrial dehydrogenase. We will be getting the purple pharmacin crystal in purple color. So when we are using the fish cell line, the color formation is very less. You can see the top 96 well plate. You can see that there is no color difference. So when we are using the same nanoparticle in the MCF cell line, you can see the color difference. So we conclude that maybe we assume that the main reason is the lower mitochondrial and dehydrogenase metabolism may be the reason for the lesser color development when we are using the fish cell lines. How we overcome it. So here only we are contradicting the earlier results being published by using the fish cell lines by analyzing the fish cell lines for the cytotoxicity by the MTT assay. So MTT assay normally will be incubating the cells with 2 hours with MTT. If we are using the 2-hour incubation, we won't get the results. When we are increasing the incubation time to 16 hours only, we are getting the required color development. So with the normal addition of, when we are increasing the concentration from 1 milligram per ml, 150 microliter, then only we are getting the color. So this is the main findings we came across in our study. This is contradicting the paper published earlier. So here you can see, as in the previous slide I mentioned that the pharmacin crystal formation. So the first picture you can see that the formation of pharmacin crystal MCF cell line. You can see the quantum of crystals formed. So the same nanoparticle, it is produced when less number of, the second picture it is showing that when we are incubating in 2 hours with less amount of MTT, you can see the number of pharmacin crystal formed. When we did the modification, increasing the time and increasing the concentration, you can see the formation of the pharmacin crystal. So these are the main finding in our study. So in addition to that one, we assess the cytotoxic effect by using the Risasurin assay also. Here are the same thing we found when we are incubating the Risasurin dye for hours, then only we are getting the required result, required result of the cytotoxic assay to be assessed. So this may be my final slide. So this is the comparison of the LC50 value, maybe IC50, one correct term may be ED50 value. So when we are assessing the ED50 value on the different cell lines, so the normal somatic cell line, it was only 15 microgram per ml. When we are using the cancerous cell line, it is only 7.55. When we are using the Oscar spleen cell line, it was 45. So we conclude that when we are comparing these 3 cell lines, what we tested in our study, so the Oscar cell line may be a potential cell line to be used for doing the cytotoxic assays for the zinc oxide nanoparticle. So what the take-home message from our study, part of the study is, so these fish cell lines require optimization. So each and every fish cell line, it may be requiring optimization for doing any cytotoxic assays. The Risazurin assay will be more better assay than the MTT assay when we are using fish cell lines for doing the cytotoxicity assays. So certain fish cell lines may exhibit sensitivity, while others may show the resistance to the metal oxide nanoparticles. So among the fish tested cell line, so OA cell line we found more suitable for these toxicity assays. Then additional cell line may be explored for gaining the deeper understanding to test the cytotoxic assays in the fish cell lines. So thank you very much. Maybe acknowledge my Director General, my DDG Fisheries, my Institute Director, and the scientists and staff of my divisions. At last, the organizers of DAA for the opportunity to present in front of you our small findings on the cytotoxic assays. Thank you very much.
Thank you very much. Thumbs up. Any questions from the audience? No? Okay, thank you very much. Very informative. Oh, so we've got one. Looking for a mic.
Congratulations, Doctor. It's very good work. Actually, it opens a new area for research and for regulatory guidelines. Because when we talk about environmental toxic things, fish are the first to face the toxins. So it is a one very good work where you show that using a realistic model, like because we are using a rat cell line. When the toxin is in water, we are want to check the toxicity of that particular in the water, but we are using fish cell line. But by opening up a new area of fish cell lines to this is a good new area of opening. And my question is like, because we have several different fish cell lines, fishes are there, and they are from the different environment. When toxins are also ending into different type of environment. When I say environment is like marine, fresh, or different type. So do you see any difference in their susceptibility? Because for why fish, mice and that cell lines are used, they are universally susceptible to all the toxins. So here you are showing within one toxin there is some variation within the, so there is a lot of work need to be done. So how do you see in future this can come?
That's why. Suppose that OECD guidelines we are seeing, they are recommending only 11 fishes. The protocol is available only for the 11 fishes. So in case of fish cell line, only one protocol is available for the RTG cell line. So that is our main concern. So when we want to test some of your chemical for its LC50 value, so which cell line, which species you have to target, so it is better to use that species cell line, maybe whether marine or brackish water or cold water. So the selection of the species will be depending upon the cell line, will be depending upon the on fish what you want to test your chemical.
Thank you.
Okay, thank you very much.
So thank you very much.
I'd like to introduce our next speaker now, DAA Haidar, who's going to present lactoferrin supplementation for barramundi, limited growth response, but potent immunity. Thank you. Welcome to the stage.
Thank you very much. Good morning, everyone. Suprabhatam. Assalamualaikum warahmatullahi wabarakatuh. My name is Devi Shahida, and I am from the Research Center for Veterinary Science, BRIN, and today I would like to present our study about lactoferrin supplementation for barramundi, limited growth response but potent for immunity. This is the outline what I'm going to talk today. First, I would like to give you some introduction and objectives, then some methods that we use, some results that we found, and key conclusion. These figures here inform us the pathway of antimicrobial resistance, or AMR, in aquaculture. And this figure here tell us that some of us already know that the antimicrobial sometimes misuse or overuse, and it accumulate in aquaculture, and it can create antimicrobial resistance, and the resistant isolates can be transfer the gene, resistant gene into pathway through human, and also to contaminate the water environment. So the AMR create twin issues. First is residues in aquatic-based product, and second is that residues in aquatic animals, and together it can create microbial resistance that can severe public health and also trade barrier regulation. Urging shift away from antibiotic, then some researchers over the world have done some effort to find alternative to antibiotic. As we can see here, there are 5 modes so far has been developed, such as using biological approach, plant, animal, chemical basis, and novel strategies. Bovine lactoferrin that we studied belong to antimicrobial peptides. Now, antimicrobial peptide bovine lactoferrin is actually bioactive glycoprotein. It has a 78 until 80 kilodalton enhanced fish growth and immune response. BLF is a promising safe substitute for antibiotic in aquaculture. It helps regulate iron metabolism, reduce disease outbreak, and improve overall fish health and survival. Interest in studying BLF is growing, especially among Asian researchers, highlighting its values in sustainable aquaculture. Baramandi is an important aquaculture community in Asia Pacific region, including Indonesia and Australia. The study on BLF supplementation on Baramandi is still limited, even though existing studies on other fish species have known or have shown promising results. Now, based on the background study, we try to evaluate the effect of BLF supplementation on Baramandi by evaluating the growth performance upon the supplementation, and also we would like to find out the response in terms of immune response by observing hematological and gut microbiota changes. This is the experimental design that we use. We conduct the research at the Gondor Research Institute, Bali. We use Baramandi about 180, and with the total length of 11 centimeters in average and body weight of 23 grams in average. We use 12 control rank with completely random design with 4 treatments and 3 replicates. We divided the fish into 4 groups, and we conduct the supplementation for 7 days or 1 week, and we continue with rearing period for another 38 days. Now we produce, I would like to explain about how we produce the BLF. We use recombinant technology, BLF protein from PET11A plasmid, transformed and expressed in E. coli BL21 host cells. And then we came out with the stock solution of 7,500 microgram per ml. We prepare the commercial pellet feed and ground into fine powder, and we divide into 4 equal portions, and we add the feed binder dissolved in 100 ml of 9% natrium chloride solution. And the feed mixture will manually repellet and dried overnight, and we stored at 4 degrees until use, and the final experiment diet contained graded levels of BLF, as I said before, divided into 4 groups. For the hematological parameters, we collect the blood sample 3 ml from counter artery using sterile syringe, and we collect them into the microtube, and we use automatic analyzer to analyze the For the histopathological examination, we collect liver, spleen, kidney, and intestine tissues, and then we processing with automatic processor, and then we stain using H&E for histopathology. For the metagenomic analysis, we collect the gut sample from group A and group control, and we do the DNA extraction, amplified and sequenced with Oxford nanopore technology. For bioinformatics, we use centrifuge classified visuals using Cornell tools. And we use additional metagenomic analysis also for group A versus group D. For the statistical analysis, we use SPSS version 25, we use one-way ANOVA, and we use classical Walsh test, and we set up the significant threshold as P less than 0.05. This is the formula we use to measure the growth hormone parameters: final length, final weight gain, specific growth rate, feed efficiency, feed convention ratio, and the survival rate. Now, I move on to some results that we found in this study. This table here tells us about the growth performance of Baramandi and the terms of each abbreviation mentioned under the table. So in this study, we found no significant difference in growth performance, feed efficiency, or survival rate of Baramandi following the supplementation. Baramandi feeding behavior reduced BLF bioavailability to the pellet rejection and nutrient leaching. The structural resistance of BLF digestion limited amino acid to release for growth. Some descriptive improvement in growth and feed efficiency was seen in high dose, but not statistically significant. Survival rate remains similar across the group, with only slight non-significant high at high dose. Overall, BLF acts more as an immunomodulator than a growth promoter or survival enhancer, with benefits likely more evident under stress or pathogen challenge. This table here tells us about the hematological value of Baramandi upon dietary of BLF supplementation. Again, because limited time, the explanation of abbreviation provided under the table. So in this study, we find macrocytic anemia and thrombocytopenia across all the group. Group B saw the most severe anemia with low red blood cells, hemoglobin, hematocrit, and reduced mean corpuscular hemoglobin concentration, while group A was the least affected. Compensatory erythropoiesis produced larger, more uniform erythrocyte. Thrombocytopenia occurred in all groups, but MPV, PDV suggested regenerative platelet response. Group D still shows low platelet volume. These findings indicate spleen and anterior kidney dysfunction affecting hematopoiesis, not linked to growth or survival, but to systemic immune modulation. Group A and C showed high lymphocyte, indicating BLF-induced immune enhancement. Group B displayed chronic infection sign with elevated monocyte and granulocyte, reflecting an inflammatory response. Overall, BLF hematological effects highlight its role as an immunomodulator rather than a growth enhancer, influencing blood cell dynamics and immune response. Now I move on to histopathology result. Our study found that variation in Baramandi intestinal condition, especially in villi length. These changes are likely influenced by dietary treatment and environmental stress. The worst intestinal condition was observed in group B, shortened villi and a lumen containing undigested feed. Possible causes include suboptimal feed quality or environmental stress impairing gut health. Now this is the histology of Baramandi renal cell. The renal histopathology saw hyperamia in group A. This progressed to hemorrhage in group B and C. Increased melanoma macropacenters were observed in all groups except for D. Findings suggest that BLF supplementation may boost immune response in Baramandi. The elevated MMSC activity and no hemorrhagic lesion at high supplementation dose. Thank you. Now, this diagram here showing the number of unique and cell bacteria genera identified in the gut microbiota of Baramandi. We found that about 1,018 genera unique to A, and the control about 348 genera. while about 112 share between A and control group. Now this is the composition of gut bacteria in the control group as well as a Sankey diagram. We found that gut bacteria in the control group was dominated by two phyla, Proteobacteria and Firmicutes. Within Firmicutes, abundant families are Streptococcaceae and Clostridiaceae. Key Firmicutes species Rhambusia, Segmentum, Ramuntia, Timorensis, and Asacospora irregularis. Within Proteobacteria, dominant families were Schwannelia and Fibrionacea. Notable species we found Schwannelia, Photobacterium, Damsele, and Vibrio ponticus. Some genera are recognized as opportunistic pathogen in aquaculture. This is the corona chart representing the taxonomic composition of bacterial communities in the gut of Baramandi of the control group. As you can see here, this dominated by Photobacterium Damsele, Vibrio including Vibrio proteus, Vibrio ponticus. Along the side present the gram-positive genera as Rhambusta sedimentarium, Clostridium dacantharis, and Tarisporobacter. Okay. Again, this taxonomic composition of gut bacteria in the highest dose lactoferrin group from sample A. We found that Proteobacteria about 63.7 kilorates dominated over Firmicutes. Predominant genera Acinetobacter, Ctenotroponas, Aeromonas, Pseudomonas, and notable species Ctenotroponas, Maltophilia, and Acetonobacter johnsoni. The rise Proteobacteria indicate selective microbial modulation by high dose lactoferrin. This shift includes the emergence of potential opportunistic bacteria. Again, this is the chloride chart representing the taxonomic composition of bacterial communities in the gut microbiota from group A. This chart illustrates that the relative abundance of the across multiple hierarchical levels. The dominant bacterial community is Proteobacteria, bacteria members of class Gamma Proteobacteria. Other notable genera including Fogacella, Comamonas, Bacillus, indicating a microbial profile with increased representation of potential opportunistic and pathogenic species. Now, from our experimental study, we came up with some key conclusions. First, BLF had no significant effect on Baramandi growth or survival. Immunological improvement observed, especially lymphocytes. Gut microbiota modulation indicates immunostimulatory potential. High-dose BLF shift microbacteria increase Proteobacteria but decrease Firmicutes. This shift promotes opportunistic bacteria proliferation. And finally, BLF effects are considered species-specific, and we need to optimize formulation and delivery. Overall, then we can conclude in one conclusion, BLF supplementation in Baramandi acts more as an immunomodulator rather than a growth enhancer. In this occasion, I would like to great thanks for the organization committee of the 12th DAA and also all our colleagues that makes this presentation possible. And I would like also thank you very much for the Rumah Program Research Organization of Health, BRIN 2022, and Budget DIPA Management Talenta BRIN, and also for the Asian Indian Science Technology Development Fund 2024-2026 for giving funds and makes this presentation possible here. Thank you very much for all of your attention. Thank you.
Thank you very much, Dewi. We're up against a time barrier, so if anyone's got any questions for her, you might catch up at lunchtime or the next tea break. So thank you very much. I'd now like to call to the stage our next presenter, Nushta Paria, on the topic of use of benzalkonium chloride as a broad-spectrum therapeutic agent in different life stages of Indian major carp, Labio ruhita.
Right. Huh? Well, distinguished chairs, coordinators to my right, I think none of them mentioned, so coordinators to the right, and of course the galaxy of aquatic animal health researchers from the different part of the world. So today I am going to talk about a very basic disinfectant. In morning even Dr. Sahu sir was mentioning, nowadays we are experiencing multiple pathogen occurrences in a single disease outcome. So developing a vaccine for each and everything would have been really, really tedious. So why we cannot think about like our age-old disinfectants which have a broad spectrum activity against probably a bacteria or oomycete. So this is so morning I think Dr. Seen also mentioning, so probably 90% of the Asian aquaculture were contributing by carps, tilapia, as well as catfishes. And there is a discussion like what are the value of those produce? $2, $3, $4 a kg probably. So thinking of CRISPR-Cas9 and other therapeutics is really, really fascinating. But whether we can really, use those, why cannot we strategize our age-old disinfectants for, for a better efficacy? So even yesterday, Mohan sir was mentioning when he was, in a 50 years journey, there are a lot of progress has been made in diagnostics. Plethora of diagnostics is available. We could detect very firmly, but in therapeutics or in a treatment, yes, still I think we need to catch up. So against this background, we are taught to see our age-old how it behaves against different pathogens, the BKC. So I think in India, we get it in $4 or $5 per liter, quite effective, quite cheap as well. So, but whenever we want to see the efficacy or therapeutic against different life stages, especially the sensitive ones, eggs and larvae, we need a standardization. We know BKC is a first-hand treatment measures is suggested to the farmers. But there are very few studies, constructive studies, how much time we should give, what is the particular concentration. There are very few studies. So our work is mostly concentrated on that. Sorry. So basically I have categorized my talk in 3 different sections. First is aim 1 and 2, where we'll try to see what are the effective dose and the duration of the BKC in different life stages. And with those standardized dose, how it can, have a therapeutic effect against Saplogonia parasitica, because we are calling it a broad spectrum. So again, one against oomycete, another against Aeromonas veraneae. So this is a kind of experimental plan we had. Whatever the experiment we have done in a carp model, that is Labiorrhea, and we have taken 3 different mode of course, all are immersion method, but based on the duration, we have categorized into 3, the prolonged bath, short immersion, and a deep treatment. So for eggs, we have taken the fertilized eggs, just fertilized, and we have taken just a hatched larvae to see the larval survival. So for prolonged bath, we have taken actually 4 different concentrations, which is normally we suggest to the farmers, starting from 0.1 to 2 ppm, and we could observe that whatever we suggest now, 0.5 ppm or 0.25 ppm, it's really very efficacious. It's give a quite good result. But if you see the, lethal effect, even the 1 ppm is quite sensitive. Even 2 ppm, all are died. So we cannot really, go for 1 or 2 ppm treatment for hatching or larval survival. Then we thought to see not prolong. In hatcheries, normally we go for a short time of immersion or so. Then we taken quite around 1 to 40 ppm of BKC. Then we have standardized for 20 minutes, and again we have seen till 5 ppm it's quite fine. And if you are practicing for 20 minutes, 5 ppm, we can really suggest for eggs and larval survival. Then this is a method actually, those who are from the, very familiar with the carps, because we need a running water system for carp hatchery. So if this treatment, deep treatment, we stop the water flow for 2 or 3 minutes, and we use BKC as a therapy. If it works, it's really very, very, suggestive to the farmers. So for that, we have started from 5 ppm to 1000 ppm, and we have seen till 10 ppm, there is no adverse effect at all for a 2 minutes deep treatment, and we can really use that for therapy. Then after the standardization, we thought we see, we have used saprolegnia for, motile juice for the challenging, and also Aeromonas veraneae. These are the experiment. I am not going into detail about this. And very, very interesting result. We could see when you have challenged the eggs with the juice pores of the saprolegnia parasitica, we could see that you could just see the PC that is for around positive control, around 50% survivability is there. And we can really, for prolonged bath annual, around 80% and over 80% survivability is there. It's really, we are using a quite cheap therapeutic, and kind of result is really encouraging. And similarly, for larvae also around prolonged bath, quite good. And just note to the IP and DP, this is actually combination of the treatment which we have standardized earlier. Prolonged plus immersion for a short period. And it's giving a fantastic result. It's around 90% survivability we are getting. Then after finishing the saprolegnia part, I thought to see how it behaves with the Aeromonas veraneae. Then again, you can just note the PC, that is around 10% survivability you got when you challenged with the Aeromonas veraneae. And very, very interesting, 95% we could see that there is absolutely the eggs were fine. And the dose we have used for Aeromonas veraneae is a very virulent pathogen we have isolated from the field conditions. Similarly, for larval survival also prolonged and immersion bath, quite around over 95% survivability we have obtained. So finishing with the larval part, then we see let's see some, how it behaves with the fingerlings. The row fingerlings could sustain up to 1 ppm quite nicely, but not more than that. We cannot really suggest a 1.5 because we have around 40% cumulative mortality. And for 5 ppm, it is absolutely within 6 hours it was getting died, animals. So then we challenged the rohu, and all the experiment whatever we have done, it's not an injection. It's all standardized in an immersion method, kind of natural. We try to mimic the natural condition. And for when we challenged with the Aeromonas veraneae and treated with 0.25 or 5 ppm, because it's really giving quite good amount of protection. Then we see, we just kind of a basic thing, we wanted to see how it is really giving, in a TPC level. Whether it is really killing the bacteria, yes. For untreated give, we can see quite good amount of TPC from the kidney, not from the gills, from the kidney. And for 0.25 ppm also, we got some, but 0.5 and 1 ppm absolutely. In some time point, we didn't get a single bacteria in there. It's really good, efficacious. And in some basic study again, we wanted to see whether some of the genes are getting regulated. For gills, we really didn't get any pattern. We really didn't get any pattern. Some variations were there, but interestingly, for kidney, all the genes were upregulated, and in the later time point, again, all genes has become stabilized. So coming to the conclusion, just to take away a message, prolonged bath with probably 0.5 ppm BKC showed increased hatching and larval survival, and similarly for 2 ppm dip for 10 to 20 ppm or 20 ppm immersion bath with 5 ppm BKC is very efficacious. And both these standardized concentration are quite good for both saprolegnia as well as Aeromonas veraneae therapeutic chance. And for laboral fingerlings, we could see that around 1 ppm BKC they can survive without any lethal effect, but not more than that. And BKC with concentration 0.25 ppm or 0.5 ppm can effectively treat the Aeromonas veraneae infection in labiorhita fingerlings. Coming to acknowledgement, I really acknowledge DG ICR and Secretary DARE, DDGA Fisheries Science, Director ICR NBFJ Lucknow, organizers, of course, Fish Health Section of Asian Fisheries Society and ICR SIBA Chennai for providing this opportunity to speak in front of you. And most precisely, I like to mention this project, All India Network Project on Fish Health. This project is actually before 5 years or 7 years, really there was no therapeutic study, work undertaken in the country. But this project really gave us a platform to work on, and presently we have something to really speak on in front of you. And thank you so much for giving me this opportunity.
Thank you very much, Santosh. Very good to see work being done on non-antibiotic microbials. It's very good. Anyone has got a question from the audience? Yes, we've got one down the front. Just got to get your mic.
Thank you very much. Have you have the information also for the effect on the developmental stages? I see that you see the percent survival and the hatching rate. So my question is, any information about the development after the treatment of this BKC?
No, sorry, we have not studied in that way actually. We have just looked onto the hatching, I mean how it is, whether it is affecting the hatching percentage or larvas. We have not go for beyond the developmental stages actually.
Okay,
Sorry to say
no
that.
problem.
Any more questions on the floor? Okay, thank you very much.
Okay,
Thank you
thanks,
very
sir.
much.
Yeah, our next presenter is Natathakul Yongai. The abstract number is FH108.
Hi everyone, I'm Ms. Nathakunyang Noi. Today I will be present on the topic granulocyte tropism and lymphocyte depletion, highlight the immunopathogenesis of tilapia leg virus infection in Nile tilapia. So first, I would like to introduce everyone to the tilapia leg virus or TILV. When tilapia infected with TILV, the mortality rate can be as high as 90%. And the external symptom, we can observe the exophthalmos, skin erosion, and hemorrhage. And the internal symptoms such as the pale liver, intestinal hemorrhage, intestinal swelling, and splenomegaly. And the research on TILV has covered many aspects, but one key finding is its impact on internal organs, especially those responsible from hematopoietic organ. And you can see in this picture, the results show the depletion of both white blood cell and red blood cell in the spleen and anterior kidney. And this result met with the author study showing that the TILV infection significantly reduced in the hemoglobin level and hematocrit, highlighting its strong effect on the fish blood system. And this lead to the hypothesis that TILV infection would cause measurable change in the blood parameter. From our experiment, we found that both hematocrit and white blood cell count decreases significantly post-infection with TILV. And this information, we then began studying PBL or peripheral blood leukocytes to see how they change after TILV infection. And for this study, our conceptual framework, we focus on the early mechanism of TILV infected in Nile tilapia. And first, we start with the hypothesis that TILV infection leads leukopenia. And this may occur through two main mechanisms. And the first one is the damage in leukopoietic organ, and the second is TILV can induce apoptosis. And we propose TILV may initially bind to PBL and then directly impact the blood system at the same time throughout of the body and damage leukopoietic organ and induce apoptosis, which eventually lead to the abnormality in the PBL and functional role of PBLs. And this gap in knowledge lead to the objective of my research. So the first one is to examine the consequence of TILV infection on the number and profile of leukocyte subpopulation. And the second is to confirm the presence of TILV in the leukocytes of the infected tilapia using flow cytometry and immunofluorescence assay or IFA. And the third one is to investigate the mechanism involved in the alteration leukocyte subpopulation of the TILV infected fish using apoptotic assay and histopathological assessment of the leukopoietic organ. So first we want to isolate the PBL using the density gradient method. After we collected the blood, we dilute blood with L15 media and dilute at 1:1 ratio and then layer on focal centrifuge and collected PBL isolation. And then we wash the PBL with PBS, centrifuge and resuspend cell platelet with PBS. And now we will have the PBL ready for the analysis. So the PBL analysis using flow cytometry to examine the distribution of leukocyte subpopulation. And the results show the significant change in the distribution of this cell, especially a clear decrease in lymphocytes and granulocytes. This change suggests the leukopenia, particularly lymphopenia, likely contributing to the immune suppression. And based on this result, we continue to detect the TILV signal in leukocytes using immunofluorescence assay or IFA. And IFA analysis to perform present to the TILV antigen in the PBL. And result, we didn't find any viral signal in the pre-infection fish, but in post-infection, we observe a clear colocalization of the viral antigen indicate in green signal with a leukocyte nuclei indicate in blue signal. This result confirming TILV infection in these cells. And next, we detect TILV signal in leukocyte subpopulation using flow cytometry. And we found TILV can bind significantly to the granulocyte, suggesting granulocytopism. And this finding suggests that this white blood cell might serve as reservoir for the virus, helping to spread it throughout the body and decrease in granulocyte in the bloodstream. Maybe because the virus used them as a carrier to transport it into hematopoietic organs like spleen and anterior kidney, which are responsible for producing blood cells. And the next part for confirm that TILV can cross viremia by TILV viral load. We found the viral load in the blood gradually increases and indicating active and progressive infection. And this highlights the dynamic nature for TILV replication and suggests that post-infection, the virus may replicate in various organs or even within blood cells. And now we know TILV can bind to the PBL, especially granulocytes, and increases viral genome level in the blood. And the next, we want to determine the virus can spread and damage into the leukopoietic organ or induce apoptosis. So we look at the hematopoietic organs or leukopoietic organs like anterior kidney and spleen for the histopathological analysis. And the result, we found the melanoma macrophage center, eosinophilic intracytoplasmic inclusion body, and lymphocyte depletion in the TILV challenge when we compare with the uninfected fish. And this finding highlights the detrimental effect of the TILV infection on the tissue integrity, particularly in hematopoietic organ. And the last one, we examine the leukocyte viability and apoptosis using immunofluorescence assay and flow cytometry. For the IFA part, we observed the signal is apoptosis and necrosis signal increases over time post-infection. And for the flow cytometry part, in TILV infected PBL, apoptotic event increases over time. And next, we analyze separate by each subpopulation. We found a decrease in lymphocyte observed in this study may be due to TILV-induced apoptosis. And however, excessive apoptosis in lymphocyte as seen in the TILV-infected cell could explain the reduction of lymphocyte and overall leukocyte count in the infected fish. And so we can now outline the infection process of TILV in Nile tilapia. When TILV infected in Nile tilapia, the virus enter into the bloodstream and bind into the PBL, especially granulocyte, and then increase viral genome level in the blood and spread throughout the body, damage in leukopoietic organ, and induce apoptosis. This led to the impaired PBL production or leukopenia into the body, and this weakened the fish immune system and compromised its ability to fight infection. In summary, our study highlights how TILV used this mechanism to evade the immune response and establish systemic infection. And this emphasized the importance of the further molecular level investigation and the development of preventive measures such as vaccine to improve the fish health and support the sustainable growth of the tilapia aquaculture industry. And if you're interested, you can read the full version. Thank you for your attention.
Yeah, thank you so much for a very good work and nice slides. Now we can take 2 quick questions.
It's a really very good work. Among the lymphocytes, whether any B or T cell tropism is there? B or T cell tropism, any such kind is there? CD4, CD8?
Sorry?
Could you get the question? Among the lymphocytes, the virus has a tropism for both the lymphocytes and granulocytes. In the lymphocytes, whether it majorly affects B cells or T cells? Can you guess it? Any change in antibody level?
So the question, I guess, if I can interpret, did you do any differentiation in the lymphocytes? So were they T lymphocytes or B lymphocytes? Or was it just a general lymphocyte count that you measured?
Sorry,
Okay. So of the different types of lymphocytes, so we've got T cells and B cells, did you measure a differential difference between the T cells and the B cells? And as a reflection of that, was there any antibody differences in the affected cell, in the affected fish? So did you measure those, I guess, is the question. Is that right?
Yeah.
Yeah, okay. Yes, yeah. So have a go. She's not. I don't know how to explain it.
in my research, we didn't study deep into the separate into the. Yes, we look at PBL and lymphocyte, monocyte, and granulocyte, but we didn't study deep into the each subpopulation. Yes, yeah.
Thank you. It's a really very good work.
Sorry, thank you for a fantastic presentation and great work. Do you have any theory about how the virus is actually homing towards granulocytes, right? It's binding to the granulocytes, you're showing it. And why is there more apoptosis of the lymphocytes and not the granulocytes? Do you have any theories about it?
Lymphocyte and...
Yeah, because you're showing apoptosis of lymphocytes, but the virus is binding to the granulocytes.
Yes, yes.
So why?
I think in my opinion and in this research, I think the granulocytes are considered to the primary target to the virus infection rather more than the lymphocyte and monocyte, because they are often to the first response to the infection, and the granulocytes such as neutrophil or in the other study are abundant in the bloodstream and are involved into the initial defense against to the pathogen. And yes, because...
So maybe it's actually the granulocytes that are going and grabbing the virus rather than virus binding to the granulocytes.
Yeah.
That's why you're finding it much more connected to the granulocytes.
Yes. Thank you.
Thank you so much, and thank you so much. We invite our next presenter, Sanjeev Kumar Manna, FHS 109.
Good morning to everybody, honorable chairs, and distinguished researchers. I will be basically not presentation, it's not pure research presentation, it is somewhat kind of discussion to raise an issue on how to control the fungal diseases. The challenges in therapeutic management of womb-associated diseases, the challenges are of different kinds. It is the scientific or technical challenge, it is the ecological challenge, and of course, it is a legal challenge. Both the three aspects are there. Now, coming to the fungal diseases of fish, these are really very important and often neglected, because we mostly focus on the viral diseases and bacterial diseases. The mortality percentage becomes, it's very high. But in fungal diseases, they occur in all forms. They occur in the enzootic form, epizootic form, and panzootic forms. Especially the EUS is a big reminder of the panzootics. And in all cases, mortality widely varies from 10 to 100% among the culture system, and definitely another, the casualty is the hatchery. There is a lots of mortality in that. In our own study, the presentation, the color of presentation in 3 fonts, the red color is our own data, and the blue color are generally from the, I have just highlighted from the other works. And in India, it causes an annual loss of around $88 billion. And among these, IMCs are very important. They are the major targets for the fungus. The fungus is very susceptible due to any reason I do not know. They are really very susceptible for this infection. In salmon and trout, it's about 10 to 50% mortality. And besides the culture system, these are all culture systems. The fungus infections are definitely much more higher in the open water resources, especially the wetlands or oxbow lakes and reservoirs. These are very important. And in my own estimate, in oxbow lakes of India, we studied 64 wetlands, and here is the mortality pattern. It's the biological loss, the mortality percentage, and the prevalence was in 88% of the wetlands, US and saprolegnia, it is in 23%, and the annual loss per hectare is around 148 for US, and saprolegnia it is 20.58. And overall loss, if I translate, on an average is coming to around $90 million per annum. It's tremendously high. It's the taxonomy. We all know here the most important part is these are all pseudo fungi. It's not true fungi. They look like fungi, but they basically belong to the protista. And protista are all in between the plantae, fungi, animalia. This is very important, and this is again important in the sense that, they look like fungus, they are biologically like plantae, but some characters of animalia, and they respond to all kinds of drugs, starting from the antibiotic to antifungal, as well as antiprotozoal. And here again, the drug developments become very tough one, because if I target this group, the animals really get affected. The toxicity comes here because they are all biologically linked. This is very important. And coming to the infection, the cysts have these kinds of hooked hairs. This is very important for the pathogenic strains. Coming to the US, there is mainly 2 kinds of diseases. One is US, another is the saprolegnia. There comes both the saprolegnia and the achlea. And in US, it's that's the major pandemic in 1980s to 1996. Mortality, that's a massive mortality up to $100 million in some countries. 4 continents were affected. At present, the disease has become enzootic, and especially the Asian countries, including the South and Southeast Asia. This is purely, the very enzootic, as well as in Africa. And if you see that, the disease patterns, they are very, very prevalent, not in the culture system, but in the oxbow lakes and the periphery areas of the reservoirs where there are lots of macrophytes. And I estimated the loss something around 83 million in oxbow lakes of India. Coming to the saprolegnia, the important disease, here in Indian wetlands, the loss is around 11 million in oxbow lakes, and in Pangasius cage culture, it causes around 11.7% mortality. It's very, very high. This parasite, this fungi typically grows, this is the growth preparation temperature, around 16 to 24. We call it the good time fungus. In temperate areas, they grow in summers, and in tropical areas, they grow in winters. It's a good time, good timer. Coming to the main topic, the management part. The management, because it's an enzootic, it's called panzootics, and it's a huge mortality. The control is very, very important in all aspects, both in the larger animals as well as in the smaller animals, or it's all it's cause massive losses, and the major part of the control is the use of chemicals. But malachite green was the mainstay, and it has an MIC of 1 to 2 ppm. It's very, very low. It's really very effective, but it has been discontinued. After its discontinuation, there is absolutely no drug, no chemical to control it. Now, if I see the two important chemicals, copper sulfate and the bronopol. Bronopol I have not studied, but generally it's an MIC of 50 to 100 or 200. It's very, very high. And copper sulfate has a MIC of 62 to 100. And there are several works, 3 or 4 works have been done to enhance the efficacy of the copper sulfate, but not much efficient. And other chemicals are not really effective. And among these, for example, boric acid. We tested the boric acid, and it's absolutely not working against this. But this boric acid is very, very important to control the saprolegnosis in toads, in amphibians. That's the major medicine. Now, and all these chemicals are used in the bath treatment, and they need continuous bath, and it's really very difficult in the larger animals to control. Coming to the drugs, the VMPs, because it looks like a fungus, some workers, including us, have worked on the estimate whether we can control by the antifungal drugs, and here is the major points. The cotrimazole has an MIC of 1 to 2. It's really very effective. There are total 3 works. Ourselves, Dr. Warillo, and another worker has worked on aphanomyces. The MIC is 1. It's really good. Fluconazole in India, in some farms we have noted, but the MIC is practically more than 500, and it's not effective at all. And if I come into the antifungal agents, there are 4 kinds of antifungals in use in all sectors, and among them azoles are the major components. There are 130 chemicals of azoles, and there are some others, not very important. Those are all human medicines, but in these azoles are widely used in plant protection. In plant protection, tebuconazole is the major consumed, and other than besides there are propioconazole and prothioconazole. And among there are 2 groups of azoles, triazoles and imidazoles, and the imidazole is the clotrimazole, but this iconazole and miconazole are also the imidazole, they are also really effective, has a MIC of 8, but no one has tested so far. And it inhibits the cytochrome P51, is the 14 alpha D methylase, and it inhibits the ergosterol synthesis of the fungi. It inhibits the cholesterol synthesis of the animal. Here it comes the toxicity. And it in our saprolegnia and umicety, here it comes the fucosterols. It inhibits the fucosterols. Now coming to, we tested the safety, bioefficacy, availability, and those kinds of the clotrimazole, and we give the bath treatment after experimental infection. It can control it can save 82% of the affected feces. It's really very promising. Coming to the bioavailability, we gave the infeed bioavailability is 150% as compared to the injection. It is as compared to the injection always. It's more or less 100% bioavailable. Now coming to the oral administration, we give the pharmacokinetic. This is the 2-compartment model, pharmacokinetic model, and by there, from this PK study, we can assess the efficacy, in vivo efficacy. Now for fungus, if you has the area under curve divided by MIC, if it is 50, it's really effective. We are getting 150. It tremendously seems to be effective. And it is a Cmax, plasma concentration is 3.3 times higher than the MIC, and it is remaining in the blood for more than 24 hours after single dose. That is, we can reduce the dose, and it has a moderate toxicity definitely. It's not very toxic. And these are the cons points, mainly the toxicity points. It causes, the altered gill morphology, hepatotic and nephrotoxicity, but not very lethal. And this is the, ALT enzyme activity. This is the control, and these are the test states. And here the serum, and it inhibits the glucose, uptake by the cells. It causes hemolysis. This is the blood picture scenario. But overall, the major point is that in the cons, it is resistant to microbial degradation. Once you give to the environment, it cannot be degraded. It has a very long persistence time. And definitely it's because of the lipophilic, it can tend to, accumulate in the environment or in the fish tissue. Coming to the global use, before if I discuss whether it can be used or not, the bulk of the antifungals are used in the plant products. For example, in the European Union, in these 12 years, 1,20,000 tons were used. Among them, 90% was for the plant protection. And for human and animal, it is only 2.5 ton in 12 years. So it is the plants, the agricultural sector consume the bulk, and from there, we are getting a lot many, antifungal drugs in the aquatic environment. Here is the report. Lot many antifungal drugs. So definitely, if I say the for and against, the sector has a need, but the question is, it is not legally approved by any country so far. It is our mindset, because it is abundantly used in the plant sector, but not in the fisheries sector. There is a scope. We have to think or ponder upon that, and there is no MRL for it. No MRL for any antifungal for animal use. But in plant science, more than 12 drugs are there. They have the MRL for the crop, for the produce, as well as for the meat and milk. Those are animals who are consuming it. So there is every scope to improve it. We can think on that. Whether the regulators can approve it for, at least for intensive aquaculture use. Thank you all.
Thank you so much. We can take some quick questions.
Yes, ma'am.
It's a very much needed study that you have carried out by looking at all the data that has been accumulated. But let me comment more on the use of these azoles. Now, though we look at fish and the fungal diseases, recalcitrant fungal diseases in humans have become a very, very, very serious issue. We have been working and publishing on recalcitrant dermatomycosis, and we find that all the azoles that you listed, actually we now have, even through the third generation azoles, resistance.
Yeah.
And regarding the use in animals, I think more than saying it should be permitted in animals also, maybe it should be restricted in its use in plants. That would be a better way to address issues that at present have not, of course, attracted attention on looking at it. But like AMR, antimicrobial resistant, microbial refers also to resistance to fungal drugs, right? So in my opinion, this extensive use in plants could probably be one of the ways that through the soil, the dermatophytes that affect the humans have become highly resistant.
Yes. There
Very
are reports.
serious problem in the health industry, okay? Coming to the alanine transferase that you showed, alanine aminotransferase. Now, very recently, I think it was just about a week ago, that there has been some cardinal research how deletion of the alanine aminotransferase gene, ALT1, in Candida albicans reduce the virulence of the organism as well as its biofilm forming ability. And therefore, one is looking at a therapeutic way of addressing by deleting that particular gene from Candida, yes, but having a drug where they would probably have Candida itself bring about immunity in the individual, but without it having the resistance as well as the biofilm forming ability. That is just a comment. But I think too much of azoles is resulting in a lot of problems. I think.
Yeah, I have basically raised the issue whether the azoles can be used at least in some way, because if I see, there are two groups azoles, triazoles and amidazoles. The amidazoles are no more used in human use. Other than the intravaginal preparation, only cautery are used. In no other cases cautery is used. It is more or less stopped. And the 3 azoles are effective. I mean the amidazoles. All triazoles are ineffective. This is the humans are using the triazoles. In plants, it is all triazoles.
Yes, yes. So, of course, one wrong does not justify another. So, we should restrict the use in plants, because through the soil, all of the soil fungi are human pathogens also, many of them. And therefore, it would be good, instead of advocating, of course, the lesser toxic azoles may be used, but not the, okay? Thank you.
In plant cells, the azole use is increasing 10% every year. It is tremendous tendency. The dosage is, global dosage is 10 milligram per square meter, 10 microgram per square meter. This is the level of use of azole in agriculture. Per square meter for 10 microgram. It is tremendously high.
Okay.
Thank
you so much, Dr. Mana.
Thanks.
Yeah, we'll go to the next presenter. Dr. Ghosi Kato, abstract number FH110. This is our last presentation of this session.
Okay, thank you very much. First of all, I would like to thank the organizers for giving me opportunity to talk about my study, induction of variable but non-culturable VVNC state in Nocardia cereoli. So first, nocardiosis in yellowtail. Nocardia cereoli is a gram-positive weakly acid-fast and non-motile actinomycete which causes nocardiosis. Yellowtail, that is the most popular cultured fish in Japan, are usually affected by this disease. The diseased fish usually show emaciation like this. The formation of granulomas in the gills, spleen, and the trunk kidney are observed in the diseased fish. And adult fish, rather than young fish, are usually affected by nocardiosis, so the disease causes heavy economic losses. This is the most interesting point of this disease because usually the fish diseases occurred when the fish is juvenile, but in this case, adult fish are usually affected by this disease. Previously, we investigated prevalence of the infection in juvenile cultured yellowtail. The upper case shows the schedule of the aquaculture of this fish species. The yearling of the fish, first year, yearling of the fish entered in the aquaculture cage. We usually use the wild captured juvenile fish for the aquaculture. And 1 year old, the second year of the cultures, the farmer usually ship the fish for the customers. We sample the fish at the May when the entry of the juvenile, and then we sample the fish one time, 5 fish per month, and we check the existence of nocardia by PCR detections and immunostaining using a paraffin section and bacterial isolation. In the PCR detection, from May, July, August, several fish are positive. Further, immunostaining, most of all fish were positive. However, no colony was isolated in the bacterial isolation. So Nocardia cereulae existed in juvenile yellowtail, but not cultured on the medium. Yeah, we cannot see, but we hypothesize that these Nocardia may be variable but non-culturable state. So logarithmic dormancy and the VVNC state of the bacteria. Logarithmic state of the bacteria, of course, they are viable and metabolically active, and they proliferate and logarithmically proliferate. And the dormant state bacteria also viable, metabolically active, but they cannot proliferate in the culture medium. And this dormant state of the bacteria turns to logarithmic state when the removal of the stress, or we say the recovery cultures. I'm sorry, dormant state bacteria caused by the stress, such as low oxygen and low nutrition and low and high temperature and so on. If this stress continues more and more, the bacteria turn to the VVNC state. VVNC state of the bacteria are viable and metabolically active, but not proliferated. And VVNC cells need plenty of factors for resuscitation, such as metabolic products of the logarithmic state bacteria, or resuscitation promoting factors coded in the bacterial genomes. So in this study, we aim to identify the condition that makes the bacteria VVNC state in vitro, and to characterize VVNC state by comprehensive gene expression analysis. This is materials and methods. Bacteria, we use Nocardia cereulae NS01 isolate, isolated from a diseased yellowtail. Culture condition was brain heart infusion, VHI broth, in 25 degree. Recovery culture were done by 1% organ medium in 25 degree. Viability tests were performed using CFDA staining. mRNA sequencing, we used the bacteria cultured in BHI for 60 days as a VVNC state, or those cultured on 1% organ medium for 10 days as a logarithmic state. And RNA extraction and the cDNA library construction after ribosomal RNA depletion. And then we use Illumina MySeq, and we analyze the data using the mapping analysis against ORF of Nocardia cereulae UTF genome, and the differentially expressed genes was detected by the Trinity software, and analyze the BLAST2Go and the KEG pathway mapping. The first, this is a growth curve and the recovery culture. We cultured bacteria in the BHI broth for 60 days. And the highest OD was observed at 30 days post-inoculation. And then the OD gradually decreased. In the 30 and the 40 days post-inoculation, you can see recovery culture, the bacteria also grown on the 1% organ medium. So the bacteria in the culture 30 and the 40 days are the dormant, should be dormancy. In the 50 days and the 60 days post-inoculations, there are no colonies on the 1% organ medium in the recovery cultures. So these data suggest that the bacteria continuously cultured in the VHI for 50 and 60 days entered VVNC state. Then we checked the viability of these bacteria. The red signals show the negative control. We completely kill the bacteria by the maybe autoclave. And the green, blue, pink, and orange, blue one, and the gray one is cultured bacteria. Of course, 40 days, 50 days bacteria are in the dormant, 10 days and 20 days bacteria in the logarithmic state. Of course, you can see the fluorescent signal shifted to the right side. In the 10 days, 33% and 10 days, 70% of the bacteria are viable. In the 30 days and the 40 days, they are dormancy. So of course, the dormant cells, of course positive for the CFDA staining, they are viable. In the 50 days and the 60 days, they didn't grow on the recovery cultured. However, they still survive. The they are positive for the CFDA staining. So we consider that the bacteria cultured for 50 days and the 60 days entered the VVNC state. Then we performed the mRNA sequencing of the logarithmic and the VVNC state. In total, 7,145 genes were detected, and the VVNC state has the 443 specific genes expressed. And the differentially expressed genes, more than 4-fold and less than 0.001 significance, we detected the 1,331 genes in VVNC and the 670 genes in the logarithmic state. You can see the clear difference between the VVNC state and the logarithmic state. This is the top 10 GO terms in the biological process. VVNC state have expressed many genes in the lipid catabolic process and the fatty acid biosynthetic process. And this is the top 15 cake pathway upregulated in VVNC state. Here also, fatty acid metabolism and the fatty acid degradation genes related to the fatty acid metabolism and the fatty acid degradation were also detected. So this is the top 15 cake pathway upregulated in the logarithmic state. The logarithmic state bacteria express many genes related to the ribosome. These data suggest that the logarithmic state produce many proteins for the proliferation. So this is a cake pathway mapping of the cake pathway, global and overview maps. Left side is the logarithmic state, and the right side is VVNC state. We can see clearly the fatty acid metabolic pathway can be seen only in the VVNC state of the bacteria. So in conclusions, Nocardia cereoli cultured at 25 degrees for 60 days in VHI broth were not recovery cultured, but still alive, indicating that they are VVNC state. VVNC state and cereoli uniquely express 443 genes and significantly upregulated 1,331 genes. GO annotation and the cake pathway analysis show that the VVNC state expressed many genes related to the fatty acid metabolism. And I am sorry, but character changes. I cannot read this one. But anyway, Nocardia cereoli can enter VVNC state, and this state may cause asymptomatic infection of the juvenile yellowtail related yellowtail in the aquaculture for aquaculture in Japan. Okay, thank you very much for your attention.
Thank you very much. Have we got any questions for our speaker? Yes, there's one over the back there. Just need a mic.
This is a very interesting work, but I wish to know that you are getting very deep insights that a bacteria which when it is entering into viable non-culturable state, so whether you could draw any conclusion that how it could be revived from your data, what you are getting, like when fatty acid pathway is getting upregulated. So whether these can be used for reviving those bacteria?
You are asking about like a fatty acid
practical
pathway?
Aspects of this one.
Why the fatty acid pathway expressed in the VVNC state?
Yeah, if it is getting upregulated, whether it can be used for reviving those bacteria?
Yeah, basically, when the bacteria enter the VVNC state, they change the metabolic pathway from the normal way to the fatty acid state to consume the fatty acid contents inside of the cells. Because the bacteria want to decrease the such kind of metabolism or like physiological something. So they first they fully consume the fatty acid in the cytosol. So this is the like a normal or the usually seen in the VVNC state.
Thank you.
Any more questions for our speaker? Yes, we've got one down the front.
Thank you. As you know, the viable non-culturable state of a microorganisms was first studied in Chesapeake Bay, where the organisms would go into this state in order to overcome and the conditions for survival and stay in the sediment, okay? Now here you're talking about the organism going into a VBNC state in live animal. That is actually the VBNC state is meant to offer a state of survival for the organism during the stressful conditions that it may encounter, be it temperature or be it any chemical treatment or be it, extremes of any of the conditions, okay? Now, finding a VBNC state in the live animals that you showed, that is juveniles, makes me wonder what could have been the stress offered to those animals for the organisms to go into a viable but non-culturable state. That's my first question. And my second would be, therefore, if it has
Can I answer? Maybe I can remember.
Yeah.
So the first question, maybe the nutrition, the lack of the nutrition is the key for the VVNC, enter to the VVNC.
Lack of nutrition to the animals?
Our hypothesis, can I say our hypothesis?
Okay.
Because previously we detected the wild yellowtail captured close to the Tokyo Bay, also positive for the bacteria and not isolated. We we went to the fishing port and do the fishing on the boat and catch 10 fish and check the positivity of the nocardia. All of the fish positive for the nocardia in PCR and immunohistochemistry. However, the bacteria are not isolated. So most of the wild fish, wild yellowtail, already infected with nocardia. And the seawater is not rich for the nutrition. And the nocardia basically living in the soil. So the coastal area, the nocardia into the marine seawater and go to Vivian Sea. The Vivian Sea bacteria caught by the yellowtail, and this bacteria basically doesn't recognize by the fish, and so the asymptomatic infection can be happened by the catch of the Vivian Sea state of bacteria.
That brings me to the point. Most of the time, the culture of microorganisms present in the seawater or present in these animals have been something standard that we have adopted, most of the time extrapolated from humans and animal microbiology studies, okay? When we carried out studies, we found that in place of, say, nutrient agar or, agar normally that is used for isolation, when we brought down the peptone level from the usual 2% to 0.2%, the recovery rate increased by 35% of the total number of bacteria that could be isolated from samples. Therefore, we call them viable non-culturable when we haven't yet cultured. It's also like calling something autoimmune until you have found a cause for it. So one way for you to confirm that they are viable, non-culturable, would be to put them in a medium with nalidixic acid and yeast extract and see the elongation of the organism where its division is arrested but it is viable when you see them under the microscope, and then calculate between dead, live by culture and the viable non-culturable, because that would give you a true estimate. Because if it is in a dormant state is different from viable non-culturable. If it is a dormant state and later becomes active, it indicates that there are factors that are allowing it to overcome the stress and come over, okay? No, just something to, because we have been working in this area, not with this organism, and that is why I just wish to say, okay?
Yeah, actually it was very hard to find the condition of the, for enter the Vivian Sea.
Definitely.
We already tried using only seawater or something, but never, most of them died.
I would understand. But you could try the experiment to show that it's viable but non-culturable by the medium that is classically used with nalidixic acid to arrest actually the division, but elongation persists with low amount of yeast extract added to the medium. Okay. I think Professor Huaishu was the first to show that. You could see.
Thank you very much.
We'll draw to a
Thank
close. And thank you, Koshi, again.